Namaskar. In this e-content, we are talking about the bacterial reproduction, transformation, transduction, and conjugation. In this e-content, in part 3, we will be discussing about the conjugation mechanism. This chapter is under the general microbiology in BSc semester 1 of the biotechnology subject. After studying this lesson, you will be able to define conjugation in bacteria, describe the process of conjugation in bacteria, explain the importance of conjugation. Students, you have already studied in part 1 and part 2 bacterial reproduction, that is transformation and transduction mechanism of bacterial reproduction, in which bacteria take free DNA from their surrounding and incorporate it into their genome. This is transformation mechanism, and bacterial DNA fragment transferred from one bacterium to another with the help of bacteriophages is the second mechanism, that is the transduction. Now we are going to discuss about the bacterial conjugation. Bacterial reproduction includes transfer of genetic material between bacterial cells, allowing them to adapt and develop, and allowing them to thrive in diverse environment. This is common in transformation, transduction, and conjugation mechanism. In this genetic material transfer, in transformation, we have discussed that the DNA present in the surrounding environment can be taken up by the bacteria and can be incorporated inside the bacterial genome. In transduction, we have studied that the DNA can be transferred from one bacterium to another bacterium with the help of bacteriophages, that is the virus. Now, here we are going to discuss about the conjugation. Conjugation is the transfer of genetic material from one bacterial cell to another, usually in the form of plasmid, with the help of pilus, that is a connection between two bacterium cells. It was described by Joshua Lederberg and Edward Tatum in 1946. This process involves the establishment of physical connection between donor and recipient cells, allowing the transfer of plasmid or sometimes chromosomal DNA. This transfer is mediated by a special appendage called the pilus, which facilitates physical contact between the cells. Here, there is a role of F factor, which is a reproductive factor. The F factor was first described in 1946 by Joshua Lederberg and Edward Tatum as the genetic element responsible for bacterial mating. This factor is actually the presence of a genetic element at the center of the process of bacterial conjugation, known as the reproductive factor, that is F factor. The F factor is a small portion of DNA that carries the genes responsible for the conjugation process and encodes the DNA transfer machinery, whose functions are the formation of the pilus, the vehicle of the DNA transfer machinery, and other related functions. The cells that contain the F factor are known as F plus cells, while those which lack these factors, they are known as F minus cells. There is the pilus formation and cell to cell contact between the two bacteria. Here you can say that one of the bacteria is bacterial F plus strain and one of the bacteria is bacterial F minus strain. F plus strain meaning by that it carries the F plus containing factor, F factor containing the DNA, that is the plasmid DNA, while in F minus strain there is no plasmid. In a bacterial population, some cells possesses the F factor, that is F plus cells, while other do not contain this F factor, they are F minus cells. During conjugation, an F plus acts as the donor and F minus act as the recipient. The process begins with the formation of a pilus, a thin hair-like appendage that extends from the surface of F plus donor cell. Pilus contains protein subunits encoded by the F factor. In this way, the donor cell extends a pilus towards the recipient cells and establishes physical contact. This physical contact, pilus interacts with the F minus receiving cell or F minus cells, bringing the two cells in close proximity. This physical relationship is necessary for the transfer of genetic material. F factor encodes a set of proteins that form a complex machinery responsible for DNA transfer. This machinery includes proteins that facilitate the opening of a small area of the donor cell's DNA, forming a single stranded DNA molecule. As DNA is unconjugated, replication occurs, generating a complementary single-stranded DNA molecule in the donor cells. Now, the DNA is transferred once the pilus mediated connection between donor and recipient cells is established. The transfer of genetic material can begin. Recipient cells via the pilus. In the recipient cells, the single stranded DNA serves as template for the synthesis of a complementary strand, forming a circular double stranded DNA molecule. The F factor initiates copy is transferred to the recipient cells while the donor remains in the donor cells. Now there is episomal replication. In some cases, the transferred DNA molecule may exist as an episome, a plasmid capable of autonomous replication within the recipient cells. Alternatively, the transferred DNA can integrate into chromosome of the recipient cell, resulting in permanent genetic change. Now, in the recipient cell, the transferred F factor can either remain as an episome, a plasmid capable of autonomous replication, or integrate into bacterial chromosome. Here, now, two bacteria, you can see that they are formed. One was F plus bacteria. It gives its own DNA to the recipient bacteria, that is the F minus cells. And here you can see that in both the bacteria, there is the presence of plasmid after conjugation. Students, some of the strains are HFR strains. Some bacterial strains have an integrated F factor in their chromosome. These strains are known to have a high frequency recombination strains. These high frequency recombination strains are called as HFR strains. During conjugation involving HFR strains, not only the F factor is transferred, but also parts of chromosomal DNA is also transferred. F plus plasmid or reproductive factor of the donor cell gets integrated into bacterial chromosome or DNA. Attachable plasmids are known as episomes. Integration is possible because some nucleotide sequence of the bacterial chromosome are compatible with the sequence of the donor cells that integrates the reproductive vector into the chromosomal chromosome, and high frequency recombination because it is recombines 1000 times higher than the normal F plus strain, and that's why they are called as HFR strains. Now, what is the importance of conjugation mechanism of bacterial reproduction where the DNA is transferred from one bacteria to another bacterium? The first one is conjugation can go for horizontal gene transfer. In other reproduction mechanism, we have seen that there is the vertical gene transfer from parents to offspring. So unlike vertical gene transfer, which occurs during reproduction, horizontal gene transfer accelerates the spread of advantageous traits in bacterial population and which is of the same population. The second one is antibiotic resistance propagation. Bacteria carrying antibiotic resistant genes on plasmids can transfer these genes to other bacteria through conjugation, leading to the rapid spread of antibiotic resistance. The third is genetic exchange. Conjugation contributes to the genetic diversity and adaptability of the bacterial population, allowing them to respond quickly to the environmental changes. Bacteria can acquire new characters such as metabolic pathways production through the uh, transfer of genes from one bacterium to another bacterium through the conjugation mechanism. And the fifth one is F factor regulation. This is an important mechanism which can be seen only in the conjugation, where a specific genes encode proteins required for pilus formation, DNA transfer machinery, and other conjugation related functions. These genes are activated in response to environmental cues and regulatory signals. So these are the importance of bacterial conjugation. If you summarize whole of the conjugation mechanism, bacterial conjugation is a process by which bacteria directly transfer genetic material, including plasmids and chromosomal DNA between cells. This process, facilitated by F factor and pilus formation, plays an important role in the spread of traits such as genetic diversity, bacterial growth, and antibiotic resistance. An understanding of the molecular and regulatory aspect of bacterial conjugation not only expands our knowledge about the bacterial conjugation, but also its practical approach for the genetic engineering and antimicrobial activities. So these are the importance of the conjugation mechanism. By studying all the three processes, that is transformation, transduction, and conjugation, we have a deep insight about the transfer of genetic material, role of the DNA, horizontal gene transfer, and other mechanisms. They have their role in the genetic engineering and the advancement of the biotechnology. So these chapters are very important. Students, you can refer textbooks and reference books. Some of the textbooks here are displayed. This textbook is Tertora, Funke, and Case. That is Microbiology and Introduction, ninth edition, Pearson Education. Some of the reference books are Alexopoulos, Mims, and Blackwell. That is Introductory Mycology, fourth edition by John and Sons. JZM, Losenzer, Golden, Modern Food Microbiology, seventh edition, CBS Publishers and Distributors, MT Martin Co. JM, Parker J. Brock Biology Microorganism 12th edition, Pearson and Benjamin Cummings, Piljar, Chan and Krieg Microbiology 5th edition, Steiner, Ingram, Wheels and Pinter, that is General Microbiology, Wheeler, Sherwood and Wolverton and Prescott. These are the Microbiology 7th edition published by McGraw-Hill Higher for higher education. So these are some of the reference books and some of the textbooks. Students, you can also refer video resources. The link is given here. You can see well, and you can go on these video resources. You can visit text resources also, genetics, microbiology, NCBI bookshelf on nih.gov, transduction and transformation, Springer link, exchange of genes, transformation, conjugation and transduction, dynamics of bacterial chromosome, Willy online library, bacterial genetics, conjugation, transduction and transformation, bacterial recombination in PubMed literature. So these are the some of the text resources. Now come to the self-assessment. What is bacterial conjugation? The options are given. You can answer. The answer is a process of gene transfer through direct cell-to-cell contact. Second question is on screen. What is an HFR cell in bacterial conjugation? The answer is cell with a high frequency of recombination. Which bacterial system is analogous to bacterial conjugation in terms of gene transfer? The answer is transposomes. In bacterial conjugation, what is the role of a plasmid? The options are given here. The answer is it carries genes necessary for conjugation process. F plasmid contain the genes necessary for the formation of dash, which are used to transfer DNA during conjugation. The options are sex pili or pilus, plasmid or F factor. The answer is sex pili or pilus. So students, you have already studied transformation and transduction in previous e-content module. Today we have discussed about the conjugation mechanism. All these three important mechanisms have their role in the various fields of the biotechnology, and these understanding will help you for further studies. Till next module, namaskar.